So, let's start this class. So, in the last class, we talked about the enzymatic or catalytic or electrocatalytic biosensor by taking example of glucose, where glucose in the presence of oxygen and by the action of glucose oxidase gets transformed into gluconic acid, and the oxygen which is present there gets consumed and forms peroxide, and the electron liberated in that process are being recorded. So, in a way, amount of oxygen which is consumed is an direct or indirect method of understanding how much glucose is present in the analyte. Now, if we look at the different electrode systems in the glucose sensors, this system relied heavily on two different electrode systems. Primarily, the first electrode system, which consisted of at least one enzyme in a capillary thin layer between the electrode and the membrane, was responsible for the conversion of the substrate to electroactive material in the presence of interfering species. This is the first electrode system. The second electrode system was sensitive to the interfering species available in the sample by subtracting the measured current from the second electrode system from the measured current from the first electrode system. Clark's patented device was successful in monitoring the glucose. So, you realize that there are two different kind of electrode systems which are functioning, which are operating to make the Clark's electrode functional. The first electrode system, which consists of at least one enzyme, in this case it is glucose oxidase, in a capillary thin layer between the electrode and the membrane, was responsible for the conversion of the substrate to electroactive material, okay, in the presence of interfering species. The second electrode system was sensitive to the interfering species, which is generated up there, available in the sample by subtracting the measured current from the second electrode system from the measured current of the first electrode system. You can back calculate the amount of glucose which is consumed. So, if you look at this whole reaction carefully, so glucose is getting converted into gluconic acid, and this conversion is a function of oxygen. So, say for example, you have 5 units of oxygen in the beginning. So, because of this reaction, the oxygen goes to say 4 units. Now, from 5 to 4 unit, there is a minus 1 unit. Now, this minus 1 unit of oxygen is transformed into, fully transformed into, assumption is fully transformed into peroxide. If that 1 unit has transformed into peroxide, this must have led to some form of electron transfer. And that electron transfer, if you could quantify the electron transfer, then you know if from 5 unit to 4 unit, this is the amount of electron transfer. So, now if you know the amount of initial amount of glucose, and if you know the final amount of glucose, then you know how many electrons have traveled in that process. So, that means your glucose concentration, reduction of the glucose concentration in the process could be quantified in terms of electron transport. So, the only challenge of the reaction is, which you will come across soon, is a sensitive enough device to measure or quantify the number of electrons. If you can quantify So, say for example, if say this leads to say X amount of glucose reduction. So, you know that X amount, if you know the amount X and you know the current, then you can back calculate it, because then you can increase the sample here, you can see, and you can put any unknown sample based on the electron transfer, you can see how much glucose is present in the sample. There is another way of doing it. The other way of doing it is that you have oxygen here, oxygen is getting consumed. Now, when we will talk in detail how the Clark's electrode was developed, that was the very first one. So, what they used to do is, initially, they were measuring the oxygen consumption. So, the oxygen consumption is a direct way of telling how much glucose is getting consumed, because glucose is getting oxidized. So, electrochemically, Clark could measure the oxygen consumption, that electrochemical measurement. So, there will be a reduction in the oxygen content. Reduction in the oxygen content means the electroactive species which they are measuring, there will be either a reduction or an increase of that. Based on that, one can measure how much glucose is getting converted. This is the fundamental basics of it, but on the face of it, it looks very simple, but it is not that simple, and we will come to that why it is not that simple. Now, talking about, so the history of in this case we are talking about exclusively at this point about glucose. So, glucose biosensor has traveled a long way since 1960s, almost 60 years it has traveled, and it has covered, currently it is in the third generation of improvisation. But then one has to understand what were those factors which called for improvement, lead to generation 2, and from generation 2 to generation 3. So, the first generation of biosensors, if we look at it, the Clark's technology was then transferred to the Yellow Spring Instrument Company and became a commercial product in 1975. This is exactly the time when microelectronics was just picking up. With a successful launch at the first glucose analyzer based on the amperometric detection, ampere means current, of hydrogen peroxide from the sample of whole bird. The so-called model was called as 23A YSI analyzer, and 23SI 23A glucose biosensors is shown out here. This is how the biosensor used to look. The sensor probe consisted of with the immobilized enzyme membrane for the Yellow Spring instruments, read on from Paulo D'Orzio's work. So, here you have the sample. Sample could be anything. It could be urine, it could be blood, and of course, there will be a lot of interfering species. It could be anything. Okay. So, now the sample, assume it contains some glucose. You have the catalytic enzyme. So, this is the polycarbonate membrane. Underneath, the glucose passed through the polycarbonate membrane, and here you have the enzymes, immobilized glucose oxidase layer. So, now this is getting transformed into gluconic acid in the presence of glucose oxidase, and this process is being orchestrated with oxygen and glucose in the presence of glucose oxidase, making gluconic acid and hydrogen peroxide. There is a cellulose acetate membrane out here, which is allowing the peroxide to pass, and the peroxide on a platinum electrode is leaving 2 electrons and becoming oxygen. So, this process out here is oxidation, whereas here oxygen to hydrogen peroxide was a reduction process. we are reversing it on the platinum electrode. This was the first generation of, so if you know the peroxide to oxygen liberation by ejection of electron, these electrons are being quantified in this whole process. Now, theory of reaction and monitoring. The YSI 23A biosensors was also based on Clark's electrode scheme. However, it relied on hydrogen peroxide oxidation for glucose monitoring, since hydrogen peroxide is also produced by enzymatic reaction at a concentration that is proportional to the glucose concentration, right? If you look at the reaction carefully, peroxide generation and glucose generation is directly proportional, right? So now, since hydrogen peroxide is also produced by the enzymatic reaction at the concentration that is proportional to glucose concentration. Briefly, for the construction of the biosensor, glucose oxidase was immobilized between two membrane layers. This is exactly what you are seeing between the two membrane layers. It was immobilized. Here the immobilization has happened. Now, the first layer was polycarbonate membrane, which was used to permit only glucose molecule to move towards the enzyme layer by blocking the many other larger substances, including enzymes and protein available in whole blood. So, you realize the challenge of the first challenge. You have to have a membrane which is exclusively allowing glucose. So, when we talk about biosensors, one of the major area of research is membranes. What kind of membranes you could use which are selectively permeable? You understand? They are selectively permeable. So, now when we use the word selectively permeable, that means it brings us to cell membrane in biology. Cell membranes are selectively permeable. They allow only finite number of chemical entities to cross that barrier. So, now the challenge is, what are those filter elements? So, this polycarbonate membrane, what you are seeing, is preventing pretty much everything which are on the way, because think of it. So, try to imagine the chemistry for a minute. So, here you have the polycarbonate membrane and here you have the glucose oxidase. So, there are a finite number of glucose oxidase and you have a finite number of glucose. Now, there are other molecules, say glucose is G, then there are molecules like X, Y, Z, P, Q. Now, all of them will be, if they kind of come here, what they will obstruct the glucose to bind to this or to come close to the enzyme. And if the glucose does not come close to the enzyme within a finite period of time, that will reduce the sensitivity of the device, and further it will increase the time of the device, because there are way too many interfering species. So, if now you look back, when I was telling you this part, interfering species, there are a lot of species which can interfere. So, this polycarbonate membrane, there are two membranes. In between the two membranes, you have the glucose oxidase which are lined up. The polycarbonate membrane preferably allows mostly the glucose moieties, but I am sure there will be leakage of other things of the same molecular weight or size or shape. Okay. So, now the first layer was polycarbonate membrane, which was used to permit only glucose molecule to move towards the enzyme layer by blocking many other larger substances, including enzymes and protein available in whole blood, thus decreasing the interference effect of the species and thereby increasing the sensitivity and reducing the time of detection. Therefore, only glucose reached the enzyme layer where it was oxidized. So, glucose again repeating for your understanding, plus oxygen in the GOX layer is getting, glucose is getting oxidized. Therefore, only the glucose reaches the enzyme layer where it is oxidized. The product hydrogen peroxide from this reaction passed through the cellulose acetate membrane, which also acted as a barrier for larger molecules. Finally, hydrogen peroxide was amperometrically detected at the platinum electrode surface. So, this is the very basic fundamental theory of how this whole process happens. Now, if you look at the reaction from electrode point of view, this part is clear to you. Now, peroxide is getting oxidized at this particular voltage on this with versus AgCl electrode. So, this is when you give a specific potential on it in order to oxidize it. You are giving it, so you are essentially removing the electrons from it by giving an opposite charge. The electrons are getting attracted. So, thereby when you are removing the electron from H2O2 minus the electron, when giving a potential of 0.7 volt, what you are getting is oxygen plus a proton and 2 electrons. The first generation biosensors of 23A sensor probe was relatively expensive due to the presence of this element, because platinum is costly. It is platinum which made it fairly expensive, and that set the tone for the next generation, prone to effect, and further presence of platinum electrode and applied high voltage. When we talk about the applied high voltage, we are talking about 0.7 volt, made the system prone to the effects of interfering species in the absence of membrane. Now, when you are putting an applied voltage of 0.7 volt, 0.7 volt leads to generation of other interfering species which gets degenerated at that particular voltage. So, there is no straightforward route that we do this reaction, this is what is going to happen, especially when we are talking about samples like urine, samples like blood, samples like soil, whatever, or body tissue. Those are exceptionally heterogeneous mixture of many compounds, and it is exceptionally challenging to figure out what will be the fate of individual entities when you are applying a voltage this high, 0.7 volt. Of course, you are getting your things done because you, by giving a positive polarity on the platinum electrode, you could oxidize the peroxide to generate oxygen and electron and protons, where you have 2 electrons and you have those 2 protons generated. But that process also leads to interfering elements from the whole analyte, because whatsoever membrane you try, these are not the biologically active semi-permeable membrane. There will be something which will pass to the site and they will always interfere. This is one challenging problem of interference from natural samples, and this is a big challenge, and this is a challenge which is with every new sample you will face, because you have no idea that what all is that mixture contains. So, you will always face this challenge. So, these challenges, so first, platinum being a very costly thing, you cannot have a point of care device with platinum or gold electrodes, it is costly. You cannot have is a That's maybe not the right statement, but definitely costly. Second, because of the high voltage, there are a lot of interfering elements which are coming, and all these made this process to be looked with more in-depth detail, so that there are much more higher sensitivity, better recognition, and faster detection. So just to summarize back, I told you that when the glucose is present in a sample, in order to reach the glucose oxidase layer, there are a lot of interfering unit in the sample itself. So there has to be a membrane which allows exclusively glucose to pass, mostly glucose to pass. And underneath you have a glucose oxidase layer. And of course, you have the oxygen coming through. So the consumption of oxygen could be one way, or in this process, whatever the reduction of oxygen leading to peroxide could be the other way, where you convert the peroxide by oxidizing it and liberating those two electrons out there and 2 protons. Okay. So this is the overall concept of the first generation of glucose sensor. So in the next class, we'll move to the second and try to move to the third generation. Thank you.